Good evening everyone. In this video I'm going to talk about the short notes which I have taken from two chapters, chapter 2 and 3. You don't need to go through these chapters, literally you don't need to go through. I was just reading it for myself, and whenever I have lots of time I go through the videos because they have the paragraphs describing how they carry out the research activity. Just out of curiosity I have gone through them. You don't need to go through them. So, chapter 2 and 3 deals with the composition of the lipofuscin. We know that in Stargardt disease, in various retinal degenerative diseases like age-related macular degeneration or maculopathies, macular degenerations, there is accumulation of the lipofuscin. So, they wanted to study on fundus fluorescence. What is the cause of this fluorescence? From which part the fluorescence is coming, and what is the composition of that molecule. They wanted to study its composition because if you know how it is formed, then you can block those formative stages. And also they have seen that as age advances, even in the normal conditions, as age advances, there is accumulation of the lipofuscin, and even under the normal conditions, with increase in age, there is increase in the fundus autofluorescence. So it is linked to the substance lipofuscin they have called. In the last video, we have seen the three things mainly responsible for the accumulation of the lipofuscin: autophagy, cellular stress, and lysosomal dysfunction. Lysosomal dysfunction means deficiency or decrease in the enzyme activity, be it because of the mutation or because of the aging. Decrease in the enzymatic activity which are responsible for the catabolism or digestion of the lipofuscin, are the substances which are responsible for formation of the lipofuscin. So even these days, like it has been a decade, just more than a decade, that fundus autofluorescence is in use, in clinical use. So we have a little limited understanding of the composition of the lipofuscin, and like total composition of the lipofuscin. We have limited idea about it and relatively limited studies as compared to the other investigating modalities. To understand the composition of the lipofuscin, they subjected lipofuscin to forced extraction and divided into three parts: chloroform soluble, methanol soluble, and methanol insoluble. So different types of the fluorophores they separated from chloroform soluble substances, fluorophores, 10 discrete fluorophores, two green, three yellow green, one golden yellow, and one orange red fluorophore. So A2E is one of the chloroform soluble lipofuscin component or fluorophore, and that is most widely studied. So lipofuscin proteomes. Proteomes means proteins. So from where these lipofuscin proteins are coming from? There are 65 cellular proteins they have discovered coming from the cytoskeletal structures. Then transduction means phototransduction proteins, proteins of metabolism, proteins related to the mitochondrial respiratory chain, ion channel proteins, and chaperones. They also wanted to know whether the lipofuscin is the consequence of the disease process or it is the cause of the disease process. So for that, they wanted to know the composition of the lipoprotein. We have already discussed about it, and still not much is known about its composition. Like the complete composition is not known, related to, especially related to its emission wide spectrum. So studies have discovered that lipofuscin is cytotoxic, and it contributes to pathogenesis in retinal degeneration such as age-related macular degeneration and Stargardt disease. So the lipofuscin is cytotoxic. It is not just a consequence of the disease process, but it contributes to the disease process being a cytotoxic. So how it is cytotoxic? We know that the A2E is most prominently studied component of the lipofuscin, and it forms from the visual cycle retinoid, and one of the precursors of this is pyridinium bisretinoid, which is a precursor of A2E. It starts A2E formation. It starts in the outer segment of the photoreceptors, and then these outer segments are engulfed by the retinal pigment epithelial cells. Then autophagy happens, autophagolysosome formation, and then this lipofuscin forms. And what happens when there is reactive oxygen species formation increased cellular stress. These proteins undergo peroxidation, lipids undergo peroxidation, and when they undergo oxidization in this way, the products formed, oxidized proteins and oxidized lipids, formed this way. These are not easily digestible by these lysosomal enzymes, so they go on accumulating in the lysosomes and they contribute to the formation of the lipofuscin. And this A to E, it increases the photosensitivity of the retinal pigment epithelium, especially to the blue wavelength, that is the short wavelength light wave, and especially ultraviolet A waves. And what happens? There is again same cellular stress increases, damage to the DNA, then formation of the reactive oxygen species, peroxidation of the lipids and proteins, etc., etc. So, not just that, the A to E, that is synthetically formed A to E, they applied to the retinal pigment epithelial cells and they found out that it increases its photosensitivity to especially the blue light, ultraviolet A light, increasing its cellular stress level. Not just that, it also activates the complement which take part in the inflammatory process. Then third thing is it increases the expression of the VEGF in the on the retinal pigment epithelium. So obviously, as A to E is component of the lipofuscin, elevated A to E is related to the cytotoxicity and inflammatory process. And if they accumulate in the cell, we have already discussed in the first chapter that it initiates the apoptotic process, that is programmed cell death. So when there is cell death, increase in the cell death, there is decrease in the cell number and decrease in the tissue function, decrease in the retinal sensitivity here in this case. They have observed that A to E emission spectrum is 562 to 575 nanometer. The range of the emission spectrum of A to E is 562 to 575 nanometer, and lipofuscin is 600 to 610 nanometer. So they have observed that this much longer wavelength emission spectrum of the lipofuscin maybe because of the fluorophore complex or the other molecules also are present in the lipofuscin which are responsible for its fluorescence and not just A to E, because it has the wide range of the emission spectrum. Emission spectrum means the light it emits. The lipofuscin accumulation occurs more in the white patients than in the black patients, and hence the age-related macular degeneration is more commonly seen in white patients than in black patients, and density of the lipoprotein is highest in the central retina. Then it is also seen that on fundus autofluorescence, the areas which show high fluorescent activity on follow-up, they're more prone to retinal atrophy and degeneration. So it's quite obvious from all these things that lipofuscin and A to E are responsible for causation of the disease and not just the consequence of the degenerative retinal chorioretinal diseases. So this is what this chapter talks about, second chapter, and it also talks about why we should study these lipofuscin components and its formation, because if we can stop the formation of the lipofuscin, or if we enhance the degradation of the lipofuscin, because it can be subjected to the subretinal space where it can be degraded. This is what they have observed in studies in animal models. So we have certain substances which are found to decrease the lipofuscin reduction in animal models. So there are certain substances or drugs which found to decrease the levels of the lipofuscin in animal models, and still experiment is going in the phase 2 trial. I think that you have to go through the entire video, but it is still being studied in human beings. It's not yet completed. The study in the human beings is not yet completed. It's still going on. So there are these substances which are successfully found to decrease the levels of the lipofuscin in animal models. Isotretinoin, then isoprenol, which is RPE65 antagonist, then ABCR negative genes, knockout, ABCR gene knockout mice, and decrease in the serum vitamin A level. Decrease in the serum means lipofuscin from where it's coming? It's coming from the outer segment of the photoreceptors, and which takes part in the visual cycle, and serum vitamin A levels takes part in the visual cycle. So vitamin A products, they are the retinal component of, they form the retinal component of the lipofuscin. So they are connected to the formation of the lipofuscin. So if we decrease the serum vitamin A level, the decrease in the lipofuscin level was observed in animal models. Then there is isotretinoin. Isotretinoin, it slows down the synthesis of 11C's retinal regeneration, and hence the regeneration of the rhodopsin. Then isotretinoin. Isotretinoin is retinal pigment epithelium 65 antagonist. So RPE65 gene, what it does, it converts the all trans retinal into the 11C's retinal, and if it is inhibited, the visual cycle is inhibited, and hence the products related to the visual cycle, products forming during the visual cycle, these are not formed, and hence lipofuscin formation is decreased. So that is the significance of the RP65 antagonist that I'm going to talk about. It's given in the chapter 3 also, and chapter 4 also. That will talk in detail the experiment while reading, and also in short when I'm going to talk about the notes on the chapter 4. ABC are negative genes. We will talk about it in detail and in short also in chapter 4. So the chapter 2 deals with the lipofuscin formation and its components, and how these are related to the inflammatory process, and how they have cytotoxic effect, and how they take part in the inflammation by activating the complement cascade or complement reactions. This is all given in chapter 2. So this all indicates that lipofuscin and A2E, the component of the lipofuscin, which is most prominent and most widely studied fluorophore component of the lipofuscin, and the A2E, precursor of the A2E is pyridinium based retinoid. So this lipofuscin is not just the consequence of the disease process, but it is cause of the disease process. I'm going to summarize the chapter 3 only in 3-4 lines. Although autofluorescence imaging has been used in the clinical practice for almost a decade, but our understanding of fundus autofluorescence is still limited. Then animal studies have played an important role in gaining insight into the matter of cause of fundus autofluorescence at cellular and molecular level. And also, animal models have helped us to understand the formative process of lipofuscin and also the components of it. The animal model studies have demonstrated that other cells beside RPE cells may produce lipofuscin. So lipofuscin is produced in the retinal pigment epithelium, not exactly in the cell cytoplasm, in the lysosomes. Obviously, the cell cytoplasm takes part in that. Everything is interrelated, but specifically lysosomes. So, however, any observations made in animal models should subsequently be validated clinically in humans. So, these experiments they have carried out in animals, but still many of them are not carried out in human beings, because obviously you have to donate your eye just for the experimental purpose. And I feel sad for the animals, because just to gain the retinal tissue, the animals have to sacrifice their eyes against their will, against their consent. And if aliens are there, and if they are killing human beings for, or killing like they're removing their parts for the experiment, then why the human beings should be, why such human beings should have problems with that? Because anyways they are using other animals for experiments. So if you can't treat the lower animals with the respect, then if advanced aliens or advanced beings, if they are using you for experiment, it's, you can't just oppose that thing if you justify such experiments. But I'm against, even it's okay to die with the disease than just to trouble animals. and treat animals cruelly. This is my opinion, but I feel sad for the sacrificed animals against their will, against their wishes. And no human being wants to be experimented by aliens in the same way. Even these lower beings, no lower being, so-called lower beings, don't want to be sacrificed like this. They don't even know what's like how they're used and what's going to happen to them. I feel sad for these animals. But anyways, scientists are doing this for the benefit of human beings, so sometimes I get confused, but I really feel sad for these animals. But I have a lot of respect for animals, and as much as possible, treat animals with respect and respect their lives. They deserve to live a very happy, healthy, peaceful, fulfilled life. I'm not talking about the viruses in such, like the animals which are non-harmful to us, and the animals which are harmful to us, we can stay away from them as long as possible. You should stay away from the violence. So that was all about the notes or summarization of chapter 2 and 3. I deliberately didn't summarize these two chapters earlier because I wanted to go through them first, and because the summary is very little. So chapter 4 I'll talk about later on. This much is enough. Bye.